Hello, everyone, and welcome to today's webinar, Addressing Common Challenges in Lentiviral Vector Clarification Using Closed, Automated, Counterflow Centrifugation System. I am Megan Pascal of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific and Sartorius. To learn more, visit thermofisher.com and sartorius.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you're having trouble seeing or hearing the presentation. I would like to now introduce our speakers. Carl Dargitz, R&D Manager, Cell and Gene Therapy Platforms, Thermo Fisher Scientific, and Magda Tomala, Manager of Testing Solutions and Analytics at Sartorius. Carl and Magda, you may now begin your presentation.
All right, well, thank you very much for the introduction. And of course, big thanks to Magda and Sartorius for being such great collaborators on this project with myself and Thermo Fisher. So starting off, I just want to take a look at the lentiviral production workflow. And of course, here we're really focused in on the clarification step because we're looking at both two different instruments that are counterflow centrifugation systems that are really well utilized when used for the clarification step here. And you can see that these are the CTS Rotea counterflow centrifugation system and the KSEP. The Rotea, of course, from Thermo Fisher and the KSEP from Sartorius. And so, of course, we're looking at the entire workflow here, and there's a lot of important steps. But as I mentioned, we're really focusing in on this clarification step because that's what these instruments can provide. And so when we focus in here, there's a couple things that we looked at much more closely to see and show how well they're able to perform for this clarification step in your lentiviral production. The first is turbidity, and of course, this is sort of a macro look at how clarified the product really is. So it's very related to just the name of the step itself, and a very important measure of how well you're separating out your cells from your viral product. The next, of course, is even more important because that's the actual infectious titer. So even if we're able to do good separation between the cells and the virus, ultimately, if there's some kind of issue in the processing or it's too harsh, then you won't have a good infectious titer once that clarification is complete, and then everything downstream will not work the way it's supposed to. So that's really critical as well. And then the other aspect, which is certainly related to the other two, is our VPC or viral production cell viability and removal. And this, like I said, plays into those other two factors, but it's really important because if we're, again, treating these cells harshly in this clarification step, then they could die and release protein or nucleic acid into the viral product, which can then have issues with infectious titer, QC, all those types of things. So what this shows is that you're able to, and what you'll see is that you're able to really keep the cells at a high viability and remove a high percentage of them, which results, of course, in that decrease in turbidity and high infectious titer. So moving on, now that we've looked a little more closely at the workflow itself, I want to highlight some of the clarification challenges that we face in these types of workflows. So the first is the risk of shear stress, during that harvest process. This can cause, cell disruption and lead to higher impurities. So again, that really ties into everything that I've spoke about so far. If you're able to treat the cells and the virus gently, then you should be able to reduce the risk of cell death and any impurities that end up in your viral product. So it's critical to have systems and instruments that are capable of doing that when you're looking at your lentiviral clarification. The next is open process steps and things with traditional batch centrifugation, which is the most common method currently used for this clarification step. So it's clear, of course, with an open step, the major risk there is, some kind of contamination from, a bacteria or a fungus or something like that. And that's always a big concern. You spend a lot of time, effort, and resources in producing these lentiviral products. And so that kind of contamination is a big risk and something that is really reduced by having a closed system and closed process for this clarification step. And then, add to that the fact that oftentimes it needs to be done in batches, which really kind of increases the total time that it takes to do that processing. And that, is another major factor. With the continuous processing of these counterflow centrifugation technologies, you can really reduce the time by not having to do these batches. In addition, so the quality of the product and any kind of lentiviral recovery loss, which could be related to the actual virus or just the infectivity of it itself, could be related to shear stress, the total processing time. So that definitely kind of loops back into those first 2 points in a lot of ways. But that's a current issue and a current challenge, and something that really needs to be continued to improve. And I think this is where we're highlighting some of the improvements these counterflow centrifugation technologies can make on the current process. And then finally, there's really limited options for ready to use and fully closed harvest technologies that will allow for production at some of these larger scales. With viral and lentiviral production, of course, you can get up to very high volumes, and so you need to be able to process many different volumes and things at higher volumes as well. And that, again, when you look at batching these things for a manual centrifuge, that can be a real problem as well. So all these are different areas where this counterflow centrifugation technologies can improve upon the current methods of traditional centrifugation. So now I just want to take a step back and talk for a moment about how counterflow centrifugation works. This here is a cross section. We're using the Rotea rotary coupling here as an example, but it's very similar technology, of course, being both counterflow centrifuges for the KSEP. And there's really no major differences from what you'll see here between the two. So this is exemplary of both systems. So as you can see here, we have the cross section, and we're looking at two forces that are interacting with one another. We have the flow rate over on the left here with the kind of gray arrows, and the G force of the centrifuge with the blue arrows. And these are acting in opposition to one another. So as your cells enter through this central cannula, you can see we have some larger cells in blue and some smaller ones in yellow. Once that happens, they enter the cone and flow out into the actual centrifuge chamber. And so this is where balancing these two forces with one another creates a fluidized and gentle cell bed. Normally, in a lot of the applications we look at, we're trying to capture these cells for a downstream purpose. But of course, in this case, with lentiviral clarification, we're looking to capture these cells and elutriate out the virus so that we can keep the cells out of that viral product. You can see that with this technology, the great thing is that you're also able, if you needed to elutriate out smaller cells. However, in this case, we're capturing all the cells, and the VPCs that we use will be fairly uniform in size, of course. So it's a matter of balancing those forces, the flow rate and the G force, with one another to create that fluidized bed, while the small lentiviral particles and media that they're suspended in will elutriate out into a collection bag, and the cells are kept here, and then they will be harvested out into a waste bag later. So this is just to show the concept of elutriation, which is what we're doing with the lentivirus. But of course, we'll be capturing all the cells in this example. And so again, this is a really powerful technology and powerful technique for lentiviral clarification. And so we'll show you some of the results from that in the coming slides. But I just want to review one more time before we go there the kind of applications of counterflow centrifugation in general. We're talking specifically again here about the lentiviral clarification, but you're able to do that cell loading, as you can see in the upper left panel here, elutrate out smaller particles or smaller things, or just separate out the supernatant from the cells, which is essentially what we're doing here since viral particles are so small. You're then able to perform a media exchange or a wash, something like that. So if you wanted to make sure we kind of get all of the lentivirus out, you can do a quick wash through the cells to make sure that there's none sort of captured in that cell bed at all. And then cell concentration, capturing those cells. So if there was some sort of need for those downstream or something like that, you can harvest those, or in this case, most likely you're just going to be using that as waste, which works perfectly well as well. So looking into the results here, the first thing that I want to highlight and that we did when we started looking at this application was to sort of optimize the settings for Rotea. And of course, they'll talk about that later. Magda will talk about this for the KSEP as well. But what we first did was look at, two major things to understand how well we were able to do this clarification. And that was just adjusting the flow rate and centrifuge settings and looking at the viability, which is normalized viability, meaning the change between the starting population of cells and what came out after the clarification, and then cell removal. So how well are we separating out the cells from the virus? You can see here, we kept the ratio between the G-force and the flow rate fairly consistent. And across the board, we're getting, well into the 90s, mostly upper 90s from a viability standpoint. So we're really not losing any viability in the cells. And then the cell removal is also quite high. Almost all the cells are ending up separated out from the virus. So what this told us is that it's a pretty robust method, and it's pretty able to be translated into multiple different flow rates. So this is good because you're able to process at a higher flow rate, which means you should be able to tackle larger volumes with that. And it also gave us the ability to, see right off the bat that you can have cells that are healthy and not dying, and therefore not adding to the impurities of your viral product, which was obviously very encouraging. So as we move on to turbidity, you can see here our pre-clarification. This is just taken, directly out of a bioreactor. Has a turbidity in terms of NTUs of over 500. And, it does vary. We've done, a fair amount of these production runs, anywhere from, around 300 to all the way up to 600, depending on the particular run. But in this case, what we're showing here is well above 500. And most of that reduction occurs right off the bat just with the Rotea clarification. So you can see just a major drop all the way down to 37. Once we do a filtration step, we see that drop even lower, down to around 12. But the benefit there, of course, is if you were to try to just directly filter, there's of course the reason we do these clarification steps. It would likely clog the filters with as much impurities as there are in the starting clarification product. So with Rotea, you're getting almost everything done with that clarification step, and makes for a very easy filtration step, which is really encouraging, and we saw this consistently occur. So as we hop over to the cell viability and recovery, again, this is indicative of the purity and the just how well the viral product is going to perform later. We'll of course go through the additional results with that. But really what we're looking at is again, normalized viability. So meaning the change in viability from prior to processing with Rotea to after, and then the recovery of cells, meaning how many of the cells we're able to keep out of that viral product. So normalized viability, you can see clearly here is almost 100%. So we're really not losing any cells to death through that process, which means it's, a gentle processing, which is great. And then by the same token, our recovery here is quite high. We saw a little bit over 100% here, which is really just a cell counting error. So what this tells you is that we're really not seeing any of the cells going out into that viral product. So that's really encouraging as well. So the cells are behaving well. And so, that again, to highlight how much, how gentle this method is on processing the cells, post viral production, these VPCs often are a lot more sensitive than they would be at the beginning. And so the fact that we're not really seeing a change in viability and we're recovering all the cells is really encouraging and tells you everything you need to know about this processing technique. So finally, moving on to the infectious titer and LV recovery. Of course, when it really comes down to it, we can look at turbidity, we can look at the cell viability and recovery, and get a lot of good information about that. But it needs to reflect the actual functional aspect of the virus, which is the infectious titer. And so you can see here that we did several runs to look at the titer. And when you compare the pre-clarification to the Rotea post-clarification, you can see there's really hardly any change at all. So the actual LV recovery for Rotea prior to filtration is virtually 100%. So we're really not losing any infectious titer between prior to Rotea and post-Rotea clarification. Of course, we do see a little bit of a drop here after filtration. That's of course, a known quantity, something we would expect here, but it's really encouraging to see that that's happening there and not through the clarification step. So with that, I think I'll move on to the next slide and hand it off to Magda from Sartorius. So thank you.
Thanks a lot, Carl. Hello, good morning and good afternoon to everybody, also from Sartorius side. Now let me introduce to you the KSEP technology, and let me also demonstrate how powerful it can be for lentiviral vector clarification. So there are a couple of challenges in lentiviral vector manufacturing. Some of them are due to the distinct properties of the lentiviral vector itself, and some of them are due to the manufacturing environment or the requirements for manufacturing. So to name a couple, I would say that the shear sensitivity of the lentiviral vector and the reduced product recovery due to loss of infectivity. These are major problems, and the KSEP counterflow centrifugation really can address these points by imparting really a low shear force while processing. It's really a very gentle technology, working in a very similar way as just described by Carl. And I think this is really the strength and the power of that system for clarification. Looking, for instance, at another challenge when it comes more to manufacturing. So there are limited options actually for ready-to-use and fully closed harvest systems to accommodate also larger production scale processes. And these points are addressed by the KSEP by an built-in scalability for development and manufacturing using one and the same system, and doing so in a closed single-use manner. So let's have a look on the key operating benefits of the KSEP. So it's the ability to process a high range of cell densities. It can retain particles very well, particles such as cells or microcarriers. The low shear environment, definitely. And to name another, I would say it's the flexible operation. So as you can see on the schematic drawn on the left side, so you see the inside of a KSEP 400 instrument and a tubing set, and you see that there are 4 rotating chambers in the KSEP. And during operation, you have the full flexibility of using 1, 2, 3, or all the 4 chambers for centrifugation. And also you have the choice, and you can define how many cycles of centrifugation you want to do using one and the same system. And this, of course, is a very important aspect when it comes to processing a high range of bioreactor volumes and looking into scaling your process, of course. So let's have a closer look on how the KSAP really works, more on a technical level. So first you would go and install the tubing and the chamber set into the hardware. You would prime the system with a suitable buffer, and then clarification can already begin. And now the typical cycles of KSAP processing start. So the first one is cell broth is loaded into the centrifugation chambers. And at this time, the cells are concentrated in the cone, while lentiviral vector is going directly into the outflow and is washed out or is separated from the cells. Afterwards, the cells are washed and lentiviral vector is further collected. And this washing step is very effective actually because the buffer can really nicely penetrate the fluidized cell bed. So the vector recovery here is really good. And in the last step, the cells that have been collected in the rotation chamber are discharged into the waste, and thereby emptying all the chambers and making them ready for another cycle of loading, washing, and discharging. So to optimize process parameters for the KSAP 400 and for our upstream material, we also did an condition screening in a very similar fashion that was described by Carl before for the Rotia. So also in our case, we tested a couple of different combinations of G-force and flow rates, and we assessed normalized viability and cell removal capacity. And you see in the graph that actually, no matter which condition we used, we obtained very high cell removal rates, above 95%, across any condition that we tested. And also the viability was maintained very nicely. Also the data, though applying different settings, so the outcome of the trial was very comparable to Rotia data. So very comparable also between the systems. And now looking at the condition 1000 G and 150 ml per minute. So in terms of output, it is very similar to the other conditions we tested. But since it contains the highest flow rate, this in fact will impact the overall processing time of how fast a whole, let's say 100-liter bioreactor batch will be clarified. So this actually is the condition that we recommend and that we also use for further trials for clarification of lentiviral vector. And this is really to be considered when you are doing also scaling studies. So, and also in a similar way as was shown for the Rotia, we see turbidity data here. So, with the KSAP, we are also reducing turbidity significantly from pre-clarified, so crude broth material. So, the major turbidity reduction happens actually in the KSAP, so during counterflow centrifugation. And when we further filter the clarified harvest, we have, we see yet a smaller but still significant reduction in turbidity. Most importantly, though, is to please remember, so both steps are important. So KSAP is really nicely pre-clarifying the material, and the filter, the membrane filter afterwards is also needed, because actually only after this step, your lentiviral vector intermediate is ready and is suitable to be used for further downstream processing. So you would want to load it on a chromatography column now, or you would want to do TFF processing, and the material is very nicely suitable for this. So in another trial, we assessed the operation with 1 or 4 chambers in the case of 400. As just explained before, we have the options to use different numbers of chambers. And when looking on normalized viability and cell removal capacity again, not only do we see hardly any difference between the 1 versus 4 chamber conditions, but also on absolute terms, these percentages look very nice and also still comparable to Rotea. Then we run a study where we were assessing the repeatability of KSAP processing. So what we did is we processed 5, or we performed 5 individual runs or cycles with the KSAP using one and the same crude culture broth containing lentiviral vector. And as you see in the graph, so across the runs, we were obtaining very comparable lentiviral vector titers. So each run performs comparable to each other. What we did afterwards, we again filtered the clarified harvest, and here we do see a titer drop, similarly as shown by Carl before for the Rotea. And this is also expected, and generally it's associated to product loss in the filter, of course. Okay, so this is a final example of KSAP performance that I am going to show it to you. So here again, we assessed the 1 versus 4 chamber operation of the KSAP to clarify lentiviral vector broth. And we see again very similar results for lentiviral vector titers in the KSAP 400 clarified harvest. Very comparable, so no difference whether you're using 1 or 4 chamber. We're also seeing hardly any difference in the titers of KSAP clarified and filtered harvest. Most importantly, and that's when you look on the graph on the right side for vector recovery. So what we did is we calculated the full mass balance across the whole unit operation. So from crude broth to KSAP clarified filtered harvest. And here we obtained vector recovery of more than 90% after filtration. And this is actually a very nice result and a very good recovery for a sensitive product such as lentiviral vector. So now bringing together Rotea and KSAP, and if you're wondering in terms of scaling, and if you're wondering what would be the most appropriate application for each of the systems, so we would say that the Rotea system can be used for low volume, scale down, and characterization studies, while the KSAP 400 is to be used to process bioreactor volumes of lentiviral vector harvest at around 1 liter to 100 liters. And please keep in mind, remember, the G-force and flow rate are the critical parameters to be optimized across the 2 systems. To conclude our webinar, I hope that we could demonstrate to you that counterflow centrifugation is an innovative technology enabling a low shear harvest of highly sensitive products such as lentiviral vector. Counterflow centrifugation allows for harvesting in a closed manner using single-use consumables and maintaining high infectious titer. And the Rotea system and the KSEP, they show indeed comparable performance, thus enabling an easy scale up and scale down of the whole process or of the clarification process. And this is the end of the slide of the presentation. Thanks a lot.